Thank you, Bill. Hello everyone. My name is Benjamin Cordier, and today I'm going to be presenting my thesis defense titled Background Prediction for Variant Detection in a Clinical Context towards Improved Next-Generation Sequencing Monitoring of Minimum Residual Disease in Hematological Malignancies. Before getting started, I want to go over kind of a quick overview of the roadmap. So I'll first be giving a fairly extensive background, and this background is actually going to focus broadly on how we detect variants and the challenges therein. And then I'll be discussing some of the methods we use and pipelines we implemented, the results of our research, and then discuss kind of some implications, limitations, and future directions. So first of all, what is a variant? We see variants when we align a sequence to a reference genome, and they can come in a wide variety of structures. So the most basic are single nucleotide variants, and these can be insertions, deletions, and mutations, as many of you know. We also see more extensive larger variants. These are longer substitutions, insertions, deletions, inversions, and duplications. And here we see them as structural variants. And while not illustrated here, we also see variants on the chromosomal level. And so these are inversions, translocations, deletions, etc. And these can occur on the chromosome arm or the entire chromosome. And for the purpose of this research, really going to be honing in on the single nucleotide variants. Now, these variants also come in two flavors of sorts, germline and somatic. And germline variants are often heterozygote, and that means they exist on a single strand of the DNA rather than the given strand and its complement. Because when we sort variants in a germline sample, because of this, we'll actually see their allele frequencies may concentrate around the 50% mark right here. In many respects, germlines represent the simplest case. In contrast, somatic variants differ because they arise spontaneously throughout the lifetime. And these are the variants and mutations most commonly associated with cancer and other tissue-specific dysregulation of the genome. Their provenance greatly influences allele frequency, and we'll see clear deviations in a sample away from the 50% VAF mark. And this can be the result of clonality and also a global reduction in purity in a sample. Somatic variants are the primary variants that I'm interested in, and so when I'm talking about variants throughout this presentation, those are what I'm going to be talking about. Now, as I noted, there are confounders of variant allele frequency. The first is purity, which we also call normal cell contamination. The second is clonality, which means there are multiple subclones in a sample. And the third is copy number, which means there are multiple copies within the cell of a given variant, away from the homozygote, heterozygote norm. And for this research, I'll be focusing specifically on purity and clonality. So what is meant by sample purity and normal contamination? Well, if we imagine that you have a patient with leukemia and you want to sample their peripheral blood, you'll take a sample, and instead of just getting the pure cancer cells and the subclones therein, you'll actually also get normal cell contamination. And so, as an example, if you have a heterozygote that's at 50% and there's 20% contamination, we might actually see it land at about 40% variant allele frequency. So this is a global reduction. Similarly, we also see clonality, and this is something that's quite unique to cancer and other more somatic-based disorders. And so this starts with a premalignant driver mutation, and what we'll see is from there is an initial clonal expansion where the genetic instability caused by the driver mutation basically starts to include new genetic variants. And if we were to sequence at this point, we might see 4 distinct subclones of the cancer. And at this point, maybe we'll actually go get a diagnosis and start treatment. Now, the first treatment is known as induction treatment and induction therapy. And it may be completely successful, but it also may actually not get all of the subclones. Some may become extinct, others may continue on, and because of the selective pressure of the treatment, may actually diversify into new subclones and pick up new variation. And at that point, we might do consolidation treatment, and this is a cycle that can repeat with added complexity, altering the clonal structure of the cancer as we go through it. And an important thing to note is that after each treatment, we're going to see reduced variant allele frequencies of these subclones. And this is because the treatment's working, but it can also represent a challenge. So why does it represent a challenge? Well, because of minimum residual disease. Minimum residual disease is defined as the post-therapy disease burden, and it's really valuable for basically finding whether a patient has the potential to relapse and or they are in a good remission, a strong remission. It also can be really valuable in determining whether we can pursue a certain precision medicine approach for a specific variant and whether what titration and dose we can use. So this is a very valuable kind of clinical measure, and we detect this in the clinic using next-generation sequencing assays oftentimes. As I mentioned, with greater treatment success, the detection of MRD becomes more challenging. The sensitivity of our next-generation sequencing assays can be about 1 in a million cells, and that means that, in theory, the detection of a malignant clone should occur at 1 in a million cells, but this is a reality that we really don't see, and this is due to background error in sequencing data. So if I can walk you through kind of a basic illustration of the variant detection problem, imagine that we have many base calls lined up on the x-axis, and then we have allele frequencies plotted on the y-axis. If we sought to simply call variants visually by allele frequency, we might say that these 4 bases on the left are probably variants. Further, we see this clear area of noise that we might call the background variation error or background error. And from there, we might say, well, aha, these other areas could be variants, but it could also be error, and that's our low confidence region. So if we wanted to create a kind of really basic visual model of how this would work, we might draw a threshold and say at about 14% variant allele frequency, anything above we would say is a true variant, and anything below we would say we're not very confident about. So what is this background error? What is it exactly? It's actually quite complex, and there's multiple types. One of them is GC content bias, which is effectively, it's an artifact of, oftentimes, of the amplification and library preparation process in the sequencing, and it co-varies heavily with coverage. Another type of error is our homopolymer errors, and what we might see is that if there's a homopolymer run of Cs in the first 4 Cs, we might actually see that the sequencer continues doing representing these Cs when in fact there's actually some Gs and so on. And this can be an error that occurs also. Now, of course, the picture is actually more complex. There are also insertion, deletion, and substitution errors, and these can be easily mistaken for those single nucleotide variants. And these co-vary with the species as well as population or homopolymer errors, GC content bias, and in fact, they also co-vary with platform error as well. And so if we want to model this error, you could say, okay, well, let's make an error profile for each platform and species, and that'll roughly help us tease it apart. But that's kind of a non-scalable approach, and it would require a lot of legwork and would require versioning of these profiles and generally just not be ideal. So what we do is we turn to background error models. And there's a number of tools that we can use to develop these models and really help get around these background errors and understand them better. So one of the most basic tools is just getting distributions of the sequences and the variation. And these distributions can be empirical from the data, or they can be theoretical distributions, such as the Bernoulli and Gaussian distributions, which are well understood. We can also use thresholding. So we can take a global threshold and put it across and say, if there's a signal past a certain point, then we say it's true variation. Or if it's below this point, we say it's not true variation. And this can be across the distribution, say at the 95% confidence. And we can also use local thresholding or dynamic thresholding, where we take local sequence context and use that to build a distribution that changes as it moves along the sequence. And we can imagine this happening, kind of up and down. Now, once we have an error model, the question would be, where do we put it? And the answer is that we currently have many variant detection pipelines, of which the GATK is actually probably the most dominant, and that stands for the Genome Analysis Toolkit. It's most commonly used with Illumina platforms, and it actually has a background error model built into it, or part of the best practices, known as the base recalibration module. It's more technically, it's called the Base Quality Score Recalibration Module, or BQSR. So how does this model work? Well, it works by correcting base quality scores rather than the actual bases themselves. It models the platform errors using known covariates, and it does this to adjust the quality scores so that the variant callers downstream in the pipeline can have better data to call their variants with. So the rationale behind recalibrating base quality scores is that if we improve their accuracy, we can improve this downstream variant calling. But there are a number of reasons why the BQSR module may actually not be appropriate for the MRD use case. For example, the MRD, or the BQSR module, was designed for the detection of high confidence variants. And that's an approach that may be less relevant in the clinical diagnostic setting, where false negatives can be more consequential. In essence, excuse me, the BQSR module may actually be too conservative. Also, the platform base calling algorithms since the development of BQSR have been known to have improved a bit, which may actually make the BQSR module less relevant. And further, while it's unlikely to be problematic, the BQSR module was originally developed for germline data, not somatic longitudinal data, which may, while the model may work, the default or recommended parameterization may actually not be optimal. So in light of these background considerations, we developed the following research questions. Is the BQSR module reducing our ability, or the ability of the GATK pipeline, to detect low-frequency variants? And also, can existing error correction software, of which there are quite a number, be leveraged to accurately model background error in somatic sequencing data within the context of the GATK pipeline, either with or without BQSR, and thus improve sensitivity for detecting somatic low-frequency variants? From there, we developed our specific aims, which was first to create a framework to select background error models and evaluate their sensitivity and specificity for the detection of low-frequency variants, and then to apply this evaluation framework developed in AIM 1 to the ICGC TCGA Dream Challenge synthetic data, which has a truth set, allowing us to benchmark, evaluate, and then recommend a best-performing background error model that we could later validate in human minimum residual disease data. So our methods, we began by defining a model selection criteria. This criteria was developed to allow for a principled approach or selection of these background error models, and it had four main goals, which was to maintain theoretical biological compatibility, compatibility with the sequencing platform, and also compatibility with the high-performance computing system, in this case, Exacloud. It also had to have relative ease of use. So what do these criteria really actually mean? The biological compatibility was quite simple. Does the model actually have a haploid or diploid assumption? If it does, it's probably not going to be fit for the somatic use case. The sequencing platform compatibility was that it had to be either Illumina or ion torrent compatible, and the model had to receive input in either BAM, SAM, FASTQ, FASTA and qual formats. And then computationally, compatible with a Unix-based operating system, expose a functional API through the command line, and it can compile correctly with the available clusters. And ease of use, which is more squishy, was that it should have clear, well-maintained current documentation, and then have relatively simple usage setup, which means minimum pre or post processing, a single error correction step, and few or no dependencies. And then finally, ideally, these would also be under an open source license. So taking this criteria, we evaluated 61 background error models, and from there it landed on 5: Bluecoo, BFC, Lighter, Basehammer, and Quorum. Of these 5, the only 3 we were actually able to implement were BFC, Bluecoo, and Lighter. Coincidentally, these error models actually all use the same error correction approach known as the k-Mer spectrum approach. So next I'll go into how that approach works and then tell you a bit about how these error models take that approach but also add their own unique flavor to it. So first, the k-Mer spectrum approach works by breaking up the reads into k-Mers. And the k-Mers are essentially a sequence of length k being defined by the user oftentimes. And so, for example, here we might have these reads, and then we might say we're going to choose a k of 7 and break them off, and now we have these k-Mers right here. Now, once we've got a lot of k-Mers, we take them from the reads, we essentially create what's called a k-Mer distribution. And on the x-axis, we have the coverage, and we have the density here. So we see that the k-Mers are roughly normally distributed. And these are what we might call the strong k-Mers, or true k-Mers. And on the left, we have low-frequency k-Mers. And these are the ones that actually may have errors in them or variation and may not be correct. And so these are often called weak k-Mers. So how does the correction work? Broadly speaking, this approach in this approach, a threshold is used to classify the strong from the weak from that distribution, and then often leveraging an edit distance measure such as Hamming distance or Levenshtein distance. For example, we may decide that all k-Mers off by a Hamming distance greater than 2 are to be considered weak, and those with an edit distance less than or equal to 1 are strong. So we have our strong k-Mers here with a distance of less than to equal to 1, and we may say we want to correct that C because we suspect it's an error. So we try G here from these strong k-Mers, we try T, and then we try A. Next, we would use a majority vote algorithm to select the correction, in this case A, and then have our corrected sequence output. So, as I mentioned, BFC, Bluecoo, and Lighter all use this approach, but they each add their own different kind of version to it, approach. So BFC is unique among the 3 because it's a non-greedy algorithm. Essentially, it may make a correction, but it also may revisit that correction if it finds upon new data that the correction may not be optimal. It focuses specifically on substitutions, and it's also the only one that was quality aware. So BFC works by first classifying the k-Mers as strong or weak by counting, and then for each read, it finds the longest substring of which the k-Mer is trusted. If no trusted k-Mers are found, it then finds the ones that are one edit distance apart, and then looks through all of those and finds those and then corrects them. Bluecoo is a little different. It's a greedy algorithm, meaning that it makes a correction and it doesn't really revisit it, and it's also not quality aware. These are the same characteristics for Lighter. Bluecoo classifies them as strong or weak by counting again, much like BFC, and then it corrects unambiguous k-Mers to strong k-Mers. So if you have a k-Mer that there's only one possible correction, it will just simply correct it to that. There's another approach it takes when it finds an ambiguous k-Mer, which it takes the k-Mer and does a read extension on it, or it takes the read and does read extension on it until it finds an unambiguous k-Mer. and then it uses a majority vote algorithm to do the correction. Also, a unique feature of BlueQ that's quite important is that it prevents or avoids false positives by verifying corrections are supported by multiple solid k-Mers. LIDAR actually takes a different approach to classifying k-Mers between weak and strong by doing random sampling and using a dynamic threshold from the k-Mer distribution. And its aim is to turn every weak k-Mer into a strong k-Mer while minimizing the edit distance. And it does not correct ambiguous weak k-Mers, so it just skips right over those. So with our error models in hand, we then sought to develop the experimental conditions for this research. So the pipeline conditions shown here were designed to be able to compare the BQSR condition and a no BQSR condition, both without an error model and in combination with an error model. So for the condition 1, we essentially have no BQSR, no error model. And this is a departure from the GATK best practices. And then we have the no error model BQSR model pipeline. And that is basically an implementation of the BQSR practices. And this condition was designed to answer the question, is BQSR actually helping or not helping in this use case? And then the second set of conditions was with the error models. So that's BFC, BlueQ, and LIDAR. And those were run both in conjunction with BQSR and without BQSR. So what did the pipeline actually look like? Well, first we downloaded the synthetic data, and this was tumor normal pairs, whole genome sequencing, ran QA/QC using MD5 checksums and FastQC, and then used SAM tools to split the BAMs by read groups. These were pre-aligned with BWA backtrack, and then sorted those read groups and converted them to FastQ, stripping out the alignment. The reason we did this is the GATK best practices actually require the use of BWA mem. So we thought it would be best to adhere to those practices. From there, we would have essentially validated FastQs by read group, and at this point, we would apply an error model if it was required. So we would run the selected background error model, or if the condition required no background error model, we'd skip that step, and then we would take the reads and we'd shuffle them to remove any bias from the previous alignment, and then align them with BWA mem by read group. From there, we would actually merge the alignment back to the original BAM files, which was comprised of multiple read groups, and then step into the GATK best practices. Mark duplicates with Picard, apply BQSR, or no BQSR depending on the condition, and then use MUTEC2 to do variant calling. From there, we would have analysis-ready VCFs. So with our VCFs, we performed an evaluation, and we did this using sensitivity, specificity, and accuracy, among other statistics. We were able to do this because the data actually had a provided truth set, and it was synthetic. In addition to these metrics, we also made sensitivity and specificity curves by tumor log odds score thresholds. And tumor log odds the tumor log odds score is defined as the log odds for a base variant in the tumor sample relative to the normal sample. And so from there, we would be able to create these curves and then compare the area under the curve to understand the model's performance. The data we used, as mentioned, was synthetic data, and it was sourced from the ICGC TCGA DREAM Challenge, or DREAM Somatic Mutation Calling Challenge. We used 3 data sets. The first represented a germline data set with 100% purity and no subclones. The second had 80% purity, or 20% normal contamination and no subclones. And then the third was 100% purity, but also had subclones in it, so 50%, 33%, and 20%. This data was whole genome sequencing, and the coverage was about 30. And the percent of positions with a coverage greater than 20x was between 76 roughly and 79%. In total, each data set had between 900 million to 1.1 billion reads, and these were all aligned to the HG19 reference genome. So what were the results? For the BQSR, no BQSR, without an error model condition, we found that the no BQSR condition actually exhibited superior performance. We also saw that there was a divergent effect in contamination, where for the contamination data set, the no BQSR, or the BQSR condition remained consistent, but kind of strangely, the no BQSR condition seemed to improve, which was not what we expected. We also saw reduced sensitivity and specificity with clonality. Then for the no BQSR conditions with the BFC error model, we saw similar performance between recalibration conditions across the board, and the BQSR set exhibited improved performance for the 20% contamination and the clonality data sets. BFC was the only error model that actually seemed to enhance the effect of BQSR. Next, we did BlueCoo, and BlueCoo was found to be a surprisingly strong performer. There was no difference between recalibration conditions. Effectively, BlueCoo seemed to mitigate the effect of BQSR entirely. And in addition, we did see reductions, although slight, in the contamination and clonality models or pipelines versus the germline. And then for lighter, we saw a difference in recalibration condition performance for sets 1 and 2 between the no BQSR and BQSR conditions, with no BQSR outperforming the BQSR condition. And again, we saw contamination and clonality reduce performance. So how do the models compare? For the germline, the no model condition performed better than BFC and lighter for both base recalibration conditions. For BlueCoo, we saw the best performance comparable, or BlueCoo exhibited the best performance, and it was comparable between the no BQSR and BQSR conditions. And here the AUCs were a 0.9731 and 0.9746 respectively. We saw a clear difference between no BQSR and BQSR in the lighter and no model conditions here. And then we also see in the germline data the first evidence that background error models can mitigate the effect of the base recalibration entirely in BFC and BlueCoo. For the 80% purity set, BlueCoo exhibited the best performance with comparable no BQSR and BQSR conditions again. The AUCs here were in 0.9610 and 0.9558 respectively. BlueCoo was also an exception in that it was the only background error model that mitigated the effects of BQSR in this data set. The no model, no BQSR condition followed BlueCoo's performance with slightly lower AUCs. For the no BQSR, it was 0.9466. Notably, BFC exhibited improved performance in concert with the BQSR module in contrast to its no BQSR condition, which we found to be somewhat surprising considering the lay of the other models. Interestingly, for BFC, even the 20% normal contamination in set 2, these AUCs were an improvement over both comparable aggregate conditions in set 1, which exhibited the 100% purity. Overall, BFC and lighter actually performed relatively poorly when we compare them to BlueCoo and the no model conditions. And again, we saw the mitigation effect in the base recalibration for the BlueCoo condition, but we did not see it in the BFC condition. So for set 3, the set with clonality, perhaps the most interesting set, the base recalibration mitigation effect was seen for all the error models, making BQSR and no BQSR conditions comparable. We also saw a large difference between the no BQSR and BQSR conditions for the no model condition, and no BQSR, again, exhibited superior performance. BlueCoo, again, exhibited the best performance overall with AUCs of 0.9335 and 0.9339 for the no BQSR and BQSR conditions, respectively. So in addition to these AUC analyses, we actually then went and broke the data into 5% variant allele frequency ranges and computed AUCs across these ranges. And this was done to see whether the AUC values or the performance changed at different variant allele frequencies. In general, this heat map is largely as we expected. We see most AUC values between mostly AUC values of about 0 in between the 50% and 100% regions right about here. We also see a tapering of AUCs here between 15 and 0%. Now, for the recalibration conditions, we don't see any specific VAF regions where BQSR seems to influence sensitivity and specificity either way. And that's to say that the difference between the BQSR and no BQSR conditions appears to arise from a cumulative effect across all variant ranges rather than any specific ranges. Interestingly, for both BlueCoo and the no model conditions, we also see a tighter clustering of performances here for BlueCoo and here for the no model. And this continues across all the data. So this was quite interesting because in contrast, lighter, which we see right here, actually extended down farther into the higher VAF regions. And this is likely an indication of the introduction of false positives by the error model, which is kind of a known caveat to their use in this context. So the AUC of sensitivity and specificity curves offers a nice overview of how each model performed overall, but if we were to implement these, we'd actually need to select a model and by the tumor lot threshold. And to do this, we basically defined the optimal threshold as being that which maximized the sum of sensitivity and specificity. So for the no model, no BQSR aggregate condition, it exhibited the highest sensitivity for sets 2 and 3, and with sensitivity of 9.472, 94.7%, and 86.9% respectively. And the BlueCoo BQSR aggregate condition exhibited the highest for set 1, which was about 94.48%. For all data sets, BlueCoo exhibited the highest specificity overall, the best combinations of sensitivity, and the best combination of sensitivity and specificity. For sets 2 and 3, this was in combination with the no BQSR, and for set 1 with the BQSR condition. So we see that right here. Specifically, for set 1, the BlueCoo BQSR aggregate with a tumor LOD score of 12 had a sensitivity of 94.48% here, and a specificity of 94.21. Where's that? Yeah, right there. Thank you. And for set 2, the BlueCoo no BQSR aggregate with a tumor LOD score of 11 had a sensitivity of 90.11% and a specificity of 94.03%. Set 3, we also saw a reduction, as we expect, in the sensitivity due to the clonality, and with a tumor LOD score of 11, the BlueCoo no BQSR aggregate condition had a sensitivity of 83.53%, but a very high specificity of 93.64%. As mentioned, set 3 is the set that exhibits clonality, which is the most relevant to our use case. And the no model, no BQSR condition exhibited the highest sensitivity for this, but it actually had a relatively low specificity. What's interesting is that BlueCoo exhibited a slightly lower sensitivity. It was lower by about 3.5%, but its specificity was much, much higher, about 13% higher. So this really highlights the trade-off between sensitivity and specificity. For low-frequency variation, we're more interested in the sensitivity, perhaps, particularly in the clinical diagnostic setting, but specificity is really important too, and with an increase of 13% over a reduction of 3.5%, I think that there's a compelling argument that that actually would be a superior model. A second heat map of sensitivity and specificity AUCs was generated for the lower variant allele frequency ranges, and for this we used 1% VAF intervals from 15% down to 0% VAF. It's important to note here that the AUC values do include false positives, and the data here, in fact, highlights this. In evaluating the truth set and really scrutinizing it closely, it was found that no variants actually exist below the 15% VAF threshold for sets 1 and 2, indicating that the AUCs for these data sets are in fact due to the correct identification of false positives, which may have been introduced by the error models. If we notice that the no model conditions for set 1 and 2 are largely clear of AUCs that are bolstered by these false positives, it seems that the background error models were the likely source of them. So, given the lack of variance in sets 1 and 2, in regard to low frequency variance, perhaps the most important set is set 3. And looking into it, there were 66 variants between the 5% and 10% VAF regions, and 10 between the 10 to 15% VAF regions. So even once we examine set 3 more closely, however, we don't actually see an apparent increase in saturation of the AUCs. So the AUCs don't seem to be higher due to the known true positive variance in the data. So in sum, it could be said that this data set does not actually really help us get at the low frequency variance quite as much as we would like. But this is discussed in the future directions, and there is a solution to this. So, to answer the question, did BQSR actually improve performance? Well, without an additional background error model, not using BQSR did actually seem to result in superior performance on this data, which interestingly was whole genome sequencing data. The superior performance was seen even with use cases which exhibited 20% contamination and clonality. And because we actually saw an improvement in performance in the Germline case as well, this kind of brings up a larger question. Does the BQSR module improve variant detection for the Germline use case? In addition, the second question, can existing models enable sensitive detection of low-frequency variants? What we found is that the pipeline without the background error model had the highest sensitivity and specificity, or the highest sensitivity for sets 2 and 3. BlueCoo did exhibit a strong balance of sensitivity and specificity across all 3 data sets, which was something that was quite interesting. And broadly speaking, the background error models, I do believe, would improve variant detection depending on the research question. There is a caveat, though, that these background error models offer a potential to introduce false positives if these models are not parameterized correctly and we don't benchmark and validate them for the use case we're interested in. So whether background error models can actually improve the detection of clinically meaningful low-frequency variants does actually remain an open question. So another thing to ask is, this presented here is the Germline best practices. And it's interesting because our research indicates that the base recalibration didn't really help the Germline use case, yet this is something that is included in the best practices. So really what this may say is that we should look at best practices with a grain of salt, make sure we adopt correct parameterization, definitely make sure that we are benchmarking, testing, and validating pipelines, and not just blindly following them. So limitations and future directions. So as mentioned, there were a number of limitations. One of them was that we used default parameterization for all the pipeline software modules unless there were specific recommendations that were made available. And some of these were in the documentation, but not all modules had these. We also used 3 error models, but there were other strong candidates. I mentioned earlier Bayes Hammer and Quorum, and Bayes Hammer in particular may serve very well for the MRD use case. As mentioned previously, the data does not contain true low-frequency variants, less than 5% variant allele frequency. And it was also generated synthetically using BAM-Surgeon. This is great because it gives us a truth set, but it's kind of an open question how well BAM-Surgeon actually really does model the true biological variation we might see in cancer, which is very complex. So future directions, one that I'm quite excited about is a fixed dilution series that we've developed with Rick Press at the KDL. That's the Knight Diagnostic Lab here on campus. And this series was created from a serial dilution of DNA from an AML pre-treatment diagnostic assay sample into a 35-day post-treatment sample from the same patient. In the sample, there are variants on three genes, IDH, NROS, and NPM1. And these dilutions were scaled between 10% and 0%. And so this is 10%, 3% minor allele frequency, 1%, 0.3%, 0.1. So these will definitely be able to get at the question of low frequency variants. And they're really nice because we also have a truth set in these. Finally, unrelated to the dilution series, we came very close to deploying Bayes Hammer and Quorum, and I think the next step will be to revisit those also and see if we can get those up and running as well. So, next steps, benchmark those additional error models, refine the current pipeline to improve sensitivity and specificity for low-frequency variant detection with the available fixed dilution series. There's also potential for a second fixed dilution series to aid in this refinement, but that's kind of an open discussion right now. Next, apply these benchmark pipelines to KDL data generated from clinical samples sourced from serial minimum residual disease assays from AML patients. There's a lot of data at the KDL that can be used for this research, and so this may be a really great way to kind of push this research forward and really understand what's going on a little better. There's also research potential for translation to clinical assays. Right now, when we do an MRD assay, we don't actually have a normal pair, and this research really required a normal pair for the truth set. So there's an area of research that we need to kind of pursue, which is how do we actually translate these findings and these pipelines into the setting where there's potentially no normal pair. And then also further explore the development of novel background error models. So this might be something to do in the future, which is adopt or take advantage of the serial nature of the MRD assays and use known variants from previous assays to help power the detection of variants or after treatment when they're at very low variant allele frequencies. So with that, I'd like to wrap up and say thank you to my thesis advisory committee. They were amazing. And the Knight Diagnostic Lab, the Beat AML initiative funded by the Leukemia and Lymphoma Society, Diane Doctor, Lynn Schwabe, Andrea Ilg, just helped me organize this so much. And then, of course, Garrett Eichelberg at the KDL helped me cull through a lot of their data and help understand what was going on and make it very, very good for this research. And then finally, my peers and the department here at DMICE. Thank you. And that, I'd like to take some questions. Josh.
What were some of the actual challenges you faced in terms of actually I guess, juggling all of these data sets and crunching them multiple times through multiple error models? I mean, that seems intensive. Was it?
Yeah, it was quite challenging. I think across all of it, it was 24 conditions. That includes the data. So what ended up happening was we built a framework, and it was actually shell scripts and a state manager to help sure we didn't double up on computations, and we kept pushing everything through. And it was done on Exacloud, and I think in total it ended up being about 200,000 computational hours. So it's quite a lot, but not as much as I've seen some other Exacloud users use. So yeah, it was challenging, but I think actually if we continue this research, we may actually update that framework and make it more robust, because right now it's a little fragile with the shell scripts and their scope management. Yeah. Any other questions? Isa. Yeah.
You told us the k-Mer one?
Yeah, the k-Mer distribution. Let me pull that up. There we go.
So the true k-Mer distribution, one thing is interesting about it is, what would you think about or explain about the extra tail on the right side as opposed to the left side of it, and how that affects some of the false positives we were talking about?
So, I think the tail there, there's probably very, very few actual k-Mers there. And it's possible that those are just, those could be sourced from, well, this is coverage from, I believe, the actual k-Mers themselves. It's possible that it's due to reads. I'm not exactly sure. I actually didn't consider it. One of the things that I will say is that the false positives in the higher regions that we saw, I don't think that the distributions we had could support the level that we saw. So I think there are more there than we could attribute to a tail there. Hi, Ted. Yeah.
So do you have any plans to contribute your framework as an open source package? Do you think it's going to be useful for other people?
So it could be. I think for now, it needs to adopt some better practices. I was very results focused when developing it. It is open source and available on GitHub right now. So you can feel free to pull it, do whatever, and yeah, see if you can get it to work for your research. I think one of the next steps might be to translate it into a language that's a little easier to work with and has better scope, something like Python maybe. But it is available, and it's I would say it's still under active development. Any other questions?